Good morning. I am Dr. Raj Narayan Gupta, Associate Professor Biochemistry. Today's topic of discussion is translation. Before we start with translation, I would like to discuss about central dogma. What is central dogma? The central dogma of molecular biology is an explanation of the flow of genetic information within a biological system. It is often stated as DNA makes RNA and RNA makes protein. This states that once information has passed into protein, it cannot get out again. The transfer of information from nucleic acid to nucleic acid or from nucleic acid to protein may be possible, but transfer from protein to protein or from protein to nucleic acid is impossible. Information here means the precise determination of sequence either of bases in the nucleic acid or of amino acid residues in the protein. When we see that how central dogma works, so the process of flow of information from DNA to protein, ultimately the protein is synthesized for functioning in a various ways, like as an enzyme it functions, as an hormone it functions. So this information, how it transfer from one to other molecules, like DNA. DNA is a genetic material, and it is having all information about the cells, what to produce as a protein. So maybe the all phenotypic characters as well as genotypic character is stored in the form of DNA. So when cell has to divide, it has to double its genetic material before its division. So when DNA replicates, so it forms DNA from DNA. So this is the process which is known as DNA replication. Similarly, when this DNA is transcribed to form RNA, means from DNA, if RNA is synthesized, then this process is known as transcription. And after transcription is completed, then mRNA is synthesized. Ultimate product of transcription is a RNA. So when mRNA is synthesized, this is a messenger RNA, and by using the codons of messenger RNA, there is a translation process takes place. So the protein of desired the protein which is required as protein for synthesis, it is encoded in the form of mRNA codons, and then after its translation, the ultimate product which is known as protein, which is synthesized. So the whole process is known as central dogma. Now in central dogma there are three things. First is replication, DNA can synthesize DNA. Transcription, DNA can synthesize RNA, and then translation, RNA can synthesize proteins. Now what is translation? The pathway of protein synthesis is called translation because the language of the nucleotide sequence on the mRNA is translated into the language of the amino acids. The mRNA is translated from its 5' end to 3' end, producing a protein synthesized from its amino terminal end to its carboxy terminal end. Now what are the components required for translation? First is amino acid, which is the ultimate unit of protein, the smallest unit of any protein. This is required as a component. Transfer RNA is required to transport these amino acids. Messenger RNA is required as the information is in the form of codons within the mRNA. Amino acyl tRNA synthetase is an enzyme which is also required, and we'll discuss it. Now functionally component, that is ribosome, which is the center of translation, which is required. Now protein factors are required. ATP and GTP as a source of energy is also required. Now what are the steps for protein synthesis? The process of protein synthesis is divided into three stages. First is initiation, second is elongation, third is termination. So first, what is initiation? So initiation of the protein synthesis involves the assembly of the components of the translation system before the peptide bond formation occurs. These components include two ribosomal subunits, mRNA, amino acyl tRNA specified by the codons in the messages, GTP and initiation factors that facilitate the assembly of the initiation complex. In prokaryotes, 3 initiation factors are known: IF1, IF2 and IF3. While in eukaryote there are at least 9 designated as ELF to indicate the eukaryotic origin. Now first is ribosome, which is known as a center of protein synthesis. So ribosomes are large complexes of protein and RNA. They consist of 2 subunits, one large and one small, whose relative size are generally given in terms of their sedimentation coefficient, or S, that is also known as Swedenborg units. The S value are determined by the shape as well as by the molecular mass. Their numerical values are strictly not addictive. So in the prokaryotes, 50S and 30S ribosomal subunits together form a ribosome with an S value of 70. So here there is a major 50 plus 30 is actually, it should be 80, but as this sedimentation unit is basically based on its size, its surface area, because when it's going to be sedimented, then the force which is applied on its surface area. So considering it one pen, so if there is a pen, then if you open this pen, there are 2 parts. There are these 2 subunits. So one is bigger one, other is a smaller one. But when we make it complex, so the organization of these 2 subunits, it will make, so some of the area of large subunit is covered inside the small subunit. So the total area is actually reduced. So the combination of 50S plus 30S RNA will make a 70S RNA, right? Now in ribosome, first is small subunit, second is large subunit. So these subunits has 2 sites, A sites and P sites. A sites stand for amino acid sites, P stand for peptide site. So the ribosome has 2 binding sites for tRNA molecules. One is A, other is P. The A site binds an incoming amino acid tRNA as directed by the codon currently occupying the site. This codon specifies the next amino acid to be added to the growing peptide chain. The P site codon is occupied by the peptidal tRNA. This tRNA carries the chain of amino acid that has already been synthesized. And E site, which is near to A site, is also there, and that is occupied by the empty tRNA. Means the codon is shifted, all this RNA ribosomal units are shifted, and the whatever site was at A site is shifted to E site. So now this free tRNA, which has transferred its amino acid to other peptidal sites, which is on P sites, and then it will get exit. So when you see the structure, this large subunit and small subunit, then there is 3 sites we can consider. One is E, P, E, A and P. So at P site there is a tRNA which has its own carried amino acid, along with whatever amino acids peptide group is transferred to the from A site, so that this A site is getting free and this will move further, and then this P site will shift to A, and then next incoming A amino acid which carried by the tRNA is coming and occupying the sites as per codon specified on mRNA. Now what is tRNA? At least one specific type of tRNA is required per amino acid. So 2 sites are important. One amino acid attachment site. So if you see this is the clover leaf like structure. So at its top there is a amino acid binding site, and its base is known as anticodon site. So this is complementary to the codons which is on mRNA. So these codons are complementary to anticodons, and then they will bind. They have the affinity to make a hydrogen bond as in nucleic acid. So when we see the structure of DNA, there is 2 strands, helical structure, and this 2 strands are maintained because of their complementary to other strand. One strand has complementary bases to other strand, so they maintain their structure. Similarly, for this RNA peptide synthesis, protein synthesis, this mRNA has codons, and the base of tRNA has anticodon, which is its upper side contains amino acid, specific amino acid, and it is decided by specific anticodons also. So which codon will sequence the amino acid, it is pre-decided as tRNA, right? Because these tRNA, they have ability to carry a specific amino acid and to recognize the codons for that amino acid. tRNA are called the adapter molecules. Now the structure has already been discussed. So there is an acceptor arm on tRNA which will bind amino acid. So once it binds, it is called as charged tRNA, and when it transfers its amino acid to the peptide chain, then it gets discharged. So function of tRNA is to transport amino acid from the system to the peptide synthesis sites, which is known as translation site. Now codon and anticodon binding is due to complementary nature, as we have discussed, and in this way when tRNA comes charged, and then this will bind to the complementary codon which is on mRNA, and then this will transfer its amino acids and to P sites to next already have the already bonded tRNA on P sites. Now initiation, in initiation what happens? How translation is initiated? The small ribosomal subunit bind to initiation factor IF3, and then this will, after binding of this will allow binding of this ribosomal small subunits to the mRNA. Initiation codons are AUG. Okay, so the small subunit IF3 complex bind to the mRNA specifically binds to the sequence AGG, right? It is known as the sign diagonal sequence, which is found in all prokaryotic mRNAs. Now once initiation factor 3 binds to this unit, this will allow binding of this complex initiation factor plus small subunit to the mRNA specific site, which is AGG and AGG, right? This is the sign diagonal sequence. So the small subunit IF3 complex scans along the mRNA until it encounters the start codons. So once it receives, it will start scanning the whole sequence of mRNA, this complex, small subunit plus IF3 complex. This will start scanning the mRNA. Wherever it finds a start codon, it will bind and start or initiate the synthesis of peptide. The small subunit IF3 complex scans along the mRNA until it encounters the start codon. The tRNA IF3 complex also bind to the start codon. This complex of the small ribosomal subunit IF3 initiator tRNA and IF2 is called the initiation complex. So there is the tRNA which carries by binding of IF2, initiation factor 2. So first tRNA, which is the initiation, which is we can say it is a component of initiation process. So this initiator tRNA comes along with the second factor IF3, initiation factor IF2. At this point, the large ribosomal subunit joins in a molecule of GTP, is hydrolyzed, and the initiation factors are released. The ribosomal complex is now ready for protein synthesis. So the energy as a factor is GTP, here it is working. So GTP is hydrolyzed into GTP and inorganic phosphate. So one terminal phosphate group is broken down, and then after broken down of this terminal phosphodiester bond, there is a release of energy which will be used by the whole initiation factors, and this energy will release IF2 from initiator tRNA, and this energy will also use to carry large subunit to bind with small subunit along with mRNA. So this will make a complete initiation complex, and IF2 is released. When the ribosome is assembled, two tRNA binding sites are created. They are designated as P and A site, as already discussed. P stands for peptidal, A stands for amino acids. So the initiator tRNA is in the P site and the A site will be filled by the tRNA with the anticodon that is complementary to the codon, as we have discussed about, that codon are on mRNA and anticodon present on tRNA, and to the codon next to start. In this case, it is the tRNA that binds proline. First tRNA which binds proline. Then when the second tRNA base pairs with the appropriate codon in the mRNA, enzyme called peptidal transferase catalyzes the formation of a peptidal bond between the two amino acid present. While breaking the bond between FMET and its tRNA, this activity is intrinsic to the 23S RNA found in the large subunit. Okay, so this 23S unit is a part of large subunit which is of 50S nature, and since the rRNA catalyzes this process, it is referred to as the riboside. Now next is, after initiation, elongation of peptide is required. So second is elongation. At this point, the whole ribosome shift over one codon. This shift requires several elongation factors not shown, and energy from the hydrolysis of GTP. The result of the shift is that the uncharged tRNA that was in the P site is ejected, and the tRNA that was in the A site is now in the P site. So the A site is free to accept the tRNA molecules with the appropriate anticodon for the next codon in the mRNA. The next tRNA base pairs with the next codon, and peptidal transferase catalyzes the formation of peptide bond between the new amino acid and the growing peptide chain. So in this way, it starts elongating, and one tRNA, charged tRNA, will carry amino acid and bind to the codons with the help of their anticodons, and the next peptide which was already there will transfer to the incoming tRNA and its amino acid. So the whole peptide is transferred. So this way one amino acid is added, and in this way the chain of protein or peptide is elongated. This same reaction is repeated several times to make a long peptide chain. After this, there is a termination process. So how the protein synthesis is terminated? After its complete synthesis, it needs to be terminated. So when a termination codon enters the A site, translation halts, because this is the codon which has no specific tRNA to bind. So if there is no specific tRNA to bind, then this process is halt. So this specific sequence of codon, which is known as termination codon. Once this whole complex is going through the all scanning and elongation of the peptide, when it achieves codons of termination type, will halt all the process of elongation, and in this way there is the whole process of translation is terminated. The release factor causes the translation complex to fall apart and cleaves the polypeptide from the final tRNA. The polypeptide product is now free to function in the cells. The mRNA molecule is now available to be translated again. Now very often more than one ribosome will translate a single mRNA at the same time. One ribosome will initiate translation, and after it moves down the mRNA a bit, another ribosome will initiate, then another, and so on. So from one molecule of mRNA, there is many molecules of protein is synthesized at the same time. For example, there is a mRNA, and at one side there is a formation of initiation complex, So it binds and it's to start scanning the whole sequence of this mRNA. At the same time, another initiation factor and other subunits of large subunit of ribosome and a small subunit of ribosome comes and bind to the same site from where it was initiated. Second initiation of translation it done, it initiated, and at the same time when it moves along, the another molecule or another component comes and bind on the initiation site. In this way, third protein chain is initiated for synthesis. So in this way, it is not necessary that from one mRNA only one protein is synthesized, but at the same time it can synthesize many molecules of the same proteins by the organization of different components of initiation. At the same time, after the differences of in few seconds, it will start new synthesis of protein. Again after few seconds, it will again start the synthesis of new protein. So in this way, it start, and once it achieve the stop codon, the process of protein synthesis is terminated. Thank you.